Okay, so hello everybody and welcome to today's seminar. It is my great pleasure to present today's invited speaker. So my colleague Marina and I had the opportunity to meet Steen at the Artificial Life Conference 2 years ago. So he's a very highly respected researcher with an impressive CV, as you will see. So we are very excited to have him here today. So Steen is professor in physics and complex systems. His work focuses on creating minimal life from non-living materials and on how new technologies change what it means to be human. He obtained a PhD in physics at the Technical University of Denmark. Since 1988, he spent 20 years at Los Alamos National Laboratory at the United States, where he was part of the group that started the artificial life research area. So he's one of the pioneers in this research area. He has also been part of the Santa Fe Institute since 1988. Then in 2004, he co-founded the European Center for Living Technology in Venice, Italy. And when returning to Denmark, he founded the Center for Fundamental Living Technology and the Institute for Science, Society and Policy. He has consulted on science and technology issues for the European Commission, Danish Parliament, Danish Department of Defense, German Bundestag, US Congress, Navajo Nation, as well as 4 private companies. And today he's going to talk about a minimal physicochemical life. So Steen, the floor is yours.
Yeah, thank you so much. It's a great pleasure for me to be here. We just came from grueling winter up in Denmark to come down here. It was 20 degrees and the blackbirds were singing, so we like it. Yeah, so the first thing I want to do, because sometimes you get stuck and people get excited and I forget it. I want to acknowledge that what I say here has been, I've had help and people have been leading me for many years. It's about 25 years of work that I'm trying to sum up here today. And these are many people that have helped and participated in a variety of ways for what I'm going to talk about here. So I've made a plan, and first I'll sort of risk my life and talk a little bit about the origins of life. I know that you guys know much more about this than I do because you come from this astrobiology institute. So I like to be corrected and schooled afterwards. But I think that since I'm here and I've worked on sort of creating life for a long time, it'll be fun to hear how my perspective on how I believe life differs from your perspective or not. And then sort of the meat of the presentation will be a presentation or a discussion of how I have tried, or we have tried for many years, to inch our way towards actually creating a minimal living system in a beaker. And then to actually get into to figure out how to make that, we will today talk about the connection between the energetics and information. And that means that we'll discuss kinetics and thermodynamics, both experimentally and theoretically. And then we're going to explore how we can transition into life. As you guys know, nobody has really created life yet, but we are many that are trying. So let's dive into sort of what we know about, or what the little I know about the origins of life. If you look at the timeline here, where we have that from the Earth formed all the way up till today, and what we are going to talk about is something all the way down here that's long time ago, before life as we know it today emerged. And if I should say how I believe one should think about it, how this happened, then we should start with a energy gradient, a gradient of free energy, where you have the possibility for having some materials that can be made to different types of things. And the reason why I believe that you have to start with energetics is because of Landauer's principle. You know, I come from physics, and we know that you cannot create, you cannot change or copy, you cannot destroy information without available free energy. And that is fundamental. I know that many people mainly connect life with information, which is, I think, completely right. But before you can even create or maintain or copy information, you need to have free energy. So that's, in my opinion, the reason, the physical argumentation for why we have to start with what we today will call a metabolism. And we all know how that goes. I mean, you have a catalyst. The catalyst determines what actually is being made. You have resources that are turned into building blocks. And the first thing that I think probably happened is that you have that some of these building blocks, they were catalysts. So you have a catalyst that, okay, well, here it goes. You have a catalyst that that then can accelerate this particular process. If you then imagine, again, we're just talking about conceptual ideas here. So then what you think about next, or at least the way I think about it, is that you have a catalyst that have combinatorial possibilities. It should be able to exist in several states so that you can think about a selection of catalysts that are in a particular state. So once you have this, then once you have this, a cofactor that is good, then this is actually the whole thing here is a catalyst, because you have this energy transducer that's able to transfer the free energy that's available in one way or the other. It can be light, it can be chemical energy. It's probably chemical energy at the origins of life. That together with this cofactor can then create building blocks. And it's important now that these two things, they will be kept together with some kind of container that self-assembles. So the building block should in part make this cofactor. In my view, there are many molecules that you can take, sort of raw molecules that can act as an energy transducer. I don't think we need to make that at the origins of life, but we need to make cofactor molecules that accelerate or controls the energy producers or energy transducers production of building blocks. And building blocks, we both need to have these container molecules as well as the cofactor molecules, because the container have to keep these two things together to form a protocell. Now, question is, how might we make such a thing in the lab? And that's been sort of my intellectual journey for many years. And the way we think about it is that there are actually many possible transitions, in my opinion, to how to actually make life. We don't know whether it's just one transition or some transitions that is somehow converged to create life as we know it. We could imagine that you create different types of life, alien life, and it could be that, you know, some molecular systems, they died out because they weren't able to really go anywhere. They were evolutionary dead ends, whereas only our ancestor was able to create life as we know it. So how could one imagine doing that? Again, I'm not here I take the freedom to not use the building materials that is believed to exist in cosmos or out in the cosmic clouds here. I can take, or we've taken the freedom to take whatever chemical or component we can use because it's very difficult to create life. And so we are not trying to address how life actually happened, but one way of creating life. And it could be that it's just one of these evolutionary dead ends that we are making. It could be it's alien life, and could of course also be that it can eventually turn into to modern life. So the central idea is this one here, where we start with energy transduction. This is again where we have some resource molecules and we have some free energy. In our system, we are using light. It's not because I think this is how it happened, but it's just easier because it eliminates some chemicals that have to deliver free energy, and that makes the system more messy. So we have free energy that can convert resources into building blocks. And it both produces a cofactor, which later will become information, as we'll see, and container molecules. So this container self-assembles, and it can then self-assemble, it can then act as a container for the energy transducer and the cofactor. You should think about having a used piece of chewing gum, and then you decorate the surface of the chewing gum with molecules that can capture light and turn light energy, photon energy into chemical energy, as well as a cofactor that can actually control how well this process is running. And a very important part of this thing is the environment. That's a little bit difficult to see out here. But when you are working with these prebiotic systems, the environment is extremely important, because these guys, these structures, they're extremely fragile, so they can easily break. And my own inspiration has been mainly from Manfred Eichen, reading some of his original papers on coupled autocatalytic components and Ilio-Prigoghine's and his ideas of dissipative structures, how self-organization can spontaneously happen as you pump systems with energy. And for Neumann, also very importantly, that you have substrate-independent functions. You can have functions, you can have life that is independent of what material it's made out of. I know not everybody agrees on that, but that's my opinion, or that's my working hypothesis. So this is, if you forget everything else, and at some point we're going to get a little technical, but please remember this. This is sort of the blueprint. That's how I think about, or we think about life. It's not me who's sort of come to this in the end. There are many people who've been involved in sort of correcting our original ideas. So the question is, how can we make such a system here? And which mechanisms would we need to get involved? You can see I start from a different side. I don't start with the molecules. I start with a concept, a systemic concept, and then we make a game where we ask the chemist which materials could actually fit into such a system. We've tried many materials that didn't work. We had to modify the system, and we eventually came up with something like this here. So choosing materials, I mean, there are many different types of energy transducers here. We just have two. We have a light-driven one. We have a energy, what's it called, a chemical energy-driven one. We can have containers made out of all kinds of materials, and we can have these cofactors that has to replicate, because as we'll see, that's actually going to act as information later on. And even though I would have loved to study or help our chemists to figure out ways to do things like this, it'll cost tens of millions of dollars or euros to get such a system up and running. So that's completely out of the question. So we are just going with what's already known. I mean, either RNA or DNA. But it's not because I think that this is how it started, but I mean, I'm open to that, but it's just that this is too expensive to make experimentally realizable. So the fundamental idea is to make our protocell. You already know how it is organized, but I view the container here. This is the used chewing gum, where we glue a cofactor in that can replicate. We'll get back to this. And then we have this energy transducers that can actually convert light energy into chemical energy. So this is our sort of the fundamental design. Now, if we go into a little more details, then we have here at the top, we have where is it here? We have this vesicle that's decorated with short DNA strands, and it could also be RNA. It's just that DNA is much easier to work with. And then we have ruthenium complexes, because these are the ones that are best studied. So that's the starting situation. Then we feed the system with resource molecules, and the resource molecules are oligomers. It is oligomers that can anchor to the surface here, and we have picolinum ester, which is it's kind of like an oily substance that will be absorbed into the membrane, and so the system grows. And then as the system grows, when we convert the oily substance into lipids, into fatty acids, we keep the energy transducer. It doesn't undergo any changes. It's something that part of the resource, both a research and a working component. And from these oligomers, we can actually ligate them, and we get an informational molecule that works. And then we get a bunch of waste molecules out also. So we're going to discuss much more in detail how it works, but I'm going to repeat this because that's something that's not so easy, and at least took us many years to figure this out. And I know that many of my collaborators, when they start to come in, they have to have this story many times. So first of all, we activate the ruthenium complex. And when the ruthenium complex is activated, that means that we send an electron from the ruthenium atom out on one of the antennas, one of the bipyridine rings. And then if you have a DNA strand right next to it, you can actually, when you have it intercalated, you can actually you can snatch an electron from a guanine such that the ruthenium is now happy. And then this activated electron can be used to do work. And it can change a lipid precursor, a picolinum ester, into fatty acid. And then you have a diffusion, a whole diffusion in the DNA. There are hundreds of or thousands of papers that document that that's possible until you hit a oxyguanine, so that the hole is repaired, because the oxyguanine can deliver an electron, and then you can have what's it called, an electron donor or hydrogen donor that can replenish the oxyguanine, and then the system can continue. Now, the other thing is you have this. So you have this charge transfer. I can't really control this one. You have this charge transfer with the DNA. Both when you produce lipids, but also when you replicate your DNA, because you have a protection group at the end of the DNA that has to be deprotected. So you have to oxidize that group, so it's released before you can actually ligate the system, and that happens here. So you are depending on how well your DNA strand acts as a charge transfer device. That will determine how fast your metabolism operates, both for the information replication and for the lipid production. So that's why it's an information molecule, because depending on the composition of this molecule, you will have a fast or a slow charge transfer. And only the ones with a good charge transfer can actually produce more lipid molecules and can make copies of themselves. So, yeah, all right, I should have waited with that. This is the whole story. You already heard the story. We have the ruthenium complex which can produce lipid, but it's actually controlled by this cofactor, and it depends on the charge transfer in the DNA. So it's a long time ago when we figured out that this actually works. You have here the time. This is the kinetics, and we start out with picolinum ester, and then we generate many different vesicles here, a different size, a variety of sizes. And there's the kinetics that's interesting. There's something happens, and we can discuss that some other time. The important thing is that there is also hydrolysis of this picolinium ester, it's an ester, so eventually in water it'll deprotect. So this is the background reaction. So we get a dramatic acceleration of the production of lipid if we use this catalyst, but we'll produce a little bit of lipid anyways because of the background reaction. So we have also done modeling on this, of course. So then now it gets a little bit technical. We had to dive in and understand what was really going on with this network, this metabolic network. And here to the left, we have the whole network for production of fatty acid. And up here to the right, we have sort of the overall reaction without any of the intermediates. We have picolinium ester, we have dihydrophenylglycine, which is a proton, sacrificial proton donor. And what we end up with is fatty acid and picolinium salt. And then we get a reduced hydrogen donor. Now, there is this part here is made by the ruthenium complex, the red part. The charge transfer is happening up here with, it doesn't point where you think it points. So up here, this is where you have the chemistry or the charge transfer in the DNA. And here you have the fatty acid production here in the green. And it actually turns out that this hydrogen donor can be reconstituted spontaneously. And all the numbers you see here is partly experimentally determined for each of the free energy changes. And we've used also DFT methods to calculate it. I would have loved to talk more about how we did all this, but I don't have time. But I'll just mention that we can actually do DFT calculations of not all the materials. We can't do it with the charged ones. And then we can then use the relationships between the different reactions, use mass action and a number of other tricks, and then we get a linear equation system, and then we can solve this equation system, and we can get all the free energies out with the boundary conditions, with all the known free energy changes we have. And we'll get back to these free energy calculations at the end of the presentation here. But let's get back to the experimental work. So once we produce these fatty acid vesicles, you can this was our first graduate student, Sarah Maurer. She was able to do a life cycle. This was without any replicating DNA. And we're just using oxyguanine as an electron donor. And we had her do what's it called, the extrusion, so that you got a cycle of a continued cycle of production of fatty acid vesicles. Then there was this question, are we just generating fatty acid that forms new vesicles out in the solution? And this is showing that no, indeed, we do actually create with budding. And many other people have done this. Now, a couple of years ago, I started to work with Sarah Mancy's group, and they've devised a number of different ways to generate or to force vesicles to divide, to have a vesicle fission or replication or whatever, or the reproduction, or whatever you want to call it. So that's a little bit of the kinetics and experimental work on that. And then I would like to give you a little bit of an idea of how our replication, our information replication is. So here we start with anchored DNA strands here with intercalated ruthenium, and then we provide these resource molecules. You can see up here. You'll get self-assembly. These things will they'll find each other. They'll match up, and then we need to do this in deprotection of this protection group with light, which is done by having the ruthenium complex deliver or steal an electron. So you have a diffusion, a hole diffusion up here. So you actually deprotect this, and then you have activated the ends here with imidazole and some other activator. And then you start out with one over here, and you get two here. And we've done a bunch of work, but all of this is not working in the lab. Many people have done have gotten DNA to replicate, which I'll show, but not on a surface yet. So that's something we're starting to do. And the problem with this template-directed ligation and replication is that you get product inhibition. But that was solved by Julie Gibbs Davis group. Julie Gibbs, her name. She's got divorced. So they actually have they make lesions and A bases. They put lesions and A bases in such that I can cut here, such that there's very little difference between when you have the hybridization energy like this and the two pieces. Once you have the hybridization energy here, there are lesions and A bases that makes the free energy actually not as low as one should think if it was perfect. So in that way, you can actually get exponential amplification. And I haven't done experiments, but we've done a bunch of simulations here, and we have now we have with Sabine Müller in Germany, in Geiswald. So we are doing experiments. So these are the experimental from FRET signals, and we have machinery to do the simulations. But it's very difficult to predict exactly what happens because this is weird. I mean, the DNA, depending on how the sequence is, then the kinetics, it has very high sensitivity. We haven't really figured out exactly how to tune that. And just to say that in a way, not in a way, but it's actually exactly the same thermodynamic mechanism as Jill Joyce did and back in 2009 in their famous RNA self-replication experiment, where you have these. Is it somewhere? Oh, here it is. So you have. Oops. Okay, I can't figure this out. So you have these you have the hammerheads such that and you have these bulges such that the ligation, you can see the ligation is right here or above here, so that there is not much difference of the free energy when it's ligated, whether it's ligated or not. And that's what you want to have so that you can actually get a replication. And just to repeat again, because I know this is not so easy, why is the DNA replication dependent of the metabolism? It is dependent of the metabolism because without the deep protection, you won't be able to replicate because you cannot ligate. So you need to have a charge transfer of the DNA that's initiated by the ruthenium complex so that you can actually replicate the DNA molecule. So that's how you put in, that's how you can characterize DNA as a informational molecule, completely different from modern life, and also different from what most people think about when they do RNA-based origins of life, where you have the conformation of the RNA that determines the catalytic ability. Here, it is a composition and its ability to do charge transfer. But again, I mean, there are several generations of this. Some of the brightest chemists in the world, they have been hammering on getting RNA replication. They haven't succeeded yet. So it's very hard. This is much easier. So again, we are trying to make it as easy as possible, even though it's still not that easy. This is just to show that we've done some work on attaching DNA to vesicles, and it's not so easy to see. Can you see the red circle here? You can see that. Can you see there's a little bit of a red circle? This is an amplification of DNA on the surface. That is in Masiaki's lab in Japan. It has not been published yet. So that's, basing this lesion-induced DNA amplification on the surface should indeed be possible. Early on, we actually did concurrent experiments where we both made vesicles, generated vesicles, and ligated the DNA, and it worked, but we weren't able to replicate it, so we couldn't publish it. I mean, it was all over the place when we did. So, but anyway, so I just want to say that I think that we can do that under the same chemical conditions. Now, there is for actually having a replication of a protocell, we both need to have replicate. We have to produce what's it called, the fatty acid vesicle container molecules. We have to replicate the DNA, but then there's the question of charge transfer. And I'm a little bit worried here, so I just need to make sure what is this? Is this one? No, this is not working. Is it? Anybody knows the time? 20 what? What? Ah, okay. Well, I am a little worried. Anyway, so there are many different ways to estimate this charge transfer, and those of you who are familiar with it will already know, but it's been done, as I said, thousands of times and published thousands of times. So, and Moser and Dutton were some of the first. And anyways, I just want to show what we have done. Since we have not done the experimental work, other people have done that. We figured out a way to actually make a kinetic model of it, where we look at the transition. If I can get it to, where is this? Is it here? We look at the kinetics of the rates from one base pair to another base pair to another base pair, and many of them are measured, and some of them are estimated with DFT calculations, with quantum ab initio calculations. And then you get what you end up with is a system of ordinary differential equations where you can follow the diffusion of a hole in the DNA sequence. And then when you know the jumping rate, then you can estimate how long time it takes before you actually get to until you reach your oxyguanine. So this is where we start up here. This is where the DNA is attached to the membrane of the vesicle, and here's where this is a bulge. We didn't use an A base here, and here's where we have the oxyguanine, and here we have how long time it takes. This is a probability that you actually get to the end. You actually get you get your hole filled with the oxyguanine, and here it's about a little less than 1/100th of a second in this particular situation. So what you get here is you can then, by looking at what we just did, you can look at charge. You can simulate a bunch of DNA molecules, and you can look at the charge transfer, and you can also look at how well they replicate, how well they do this lesion-induced DNA amplification. And there's 160 sequences. Why 160? Because we couldn't find in the literature more than a certain number of transition rates. And then we made all the sequences we could where we knew the transition rate from each of the base pairs. So that's why we end up with 160. We have to calculate that many times, but it is 160. Many of us have done it. So anyways, and then we get to a really, in my opinion, really interesting chart. If you then, again, this is a logarithm. So this is, if you look at the charge transfer, and then you see, you plot that out here, and then you look at the logarithm to the replication rate up there. And then you get a whole bunch of simulations. All of them are all these colored dots. I'll get back to what the colors means in a second. You can plot all the DNA sequences in here, both as they work as charge transfer and as they work for replication. And we know that if we want to have the charge transfer to work, it has to be such that sorry, it's up here. The charge transfer has to be the rate limiting reaction for this process is the activation of the ruthenium complex. It has a rate of about 0.1 per second or something, so it's not very fast. So is that true? No, it's about 1 per second. So we need to have a rate that's much faster than the slowest rate in the metabolic process. So we've said that it has to be 50. The rate has to be 50 per second, which makes it faster than the photoactivation. And for the replication, we know that when we activate the DNA, for them to ligate, if you use a imidazole or something, then it has a certain lifetime in water, because water also deactivates it. So we've decided, or we took it 1 order of magnitude faster than hydrolysis. So we say that the replication rate has to be 10 to the minus 4th per second. So that means if you have a charge transfer rate that's faster than these, what's it called, 50 per second, and hydrolysis, replication rates that faster than 10 to the minus 4th per second, then you should be in the good. So in principle, if everything else is equal, then these in here, these strands in here, they would be able to generate offspring that's faster than the decay. So they should be viable. Of course, we have not, this is everything else being equal. We have made superposition there. I'm sure there's going to be nonlinear interactions. So we haven't done a full simulation where everything is interacting with everything else. So we can, now maybe this is crazy, but it was really exciting. Long time ago, we figured out that if you have a system where you have a replication of information together with reproduction of the container, you needed to the composed growth rate will be proportional to this, and we could then order all the fitness. We can order all the rate constants we saw from these 160 sequences we see here, and we can then we know this is the ones where we have a very short distance between where the ruthenium complex is very close to the oxyguanine, so they're probably not interesting when it comes to it. But anyway, so you can actually get a fitness function which we have organized in this way. Well, another way we can characterize it is by looking at these 160 strands and say, okay, which ones are good enough if you look at the functional information for the metabolic charge transfer, and you actually get that you can get, this is shown here, and this is the one for the replication here. And we just need, if you have 160 strands and they have a length of these 18 base pairs, we just need 1.34 bits of information to make sure that you are in the viable area. Again, this is just a sort of a mathematical play, but still, it gives us an idea of how one should look at the onset to life. So if we have a system as I described, and we randomly pick strands from these 160 DNA strands, then some of them the system will just die. But then at some point you'll get a strand that brings the system above the viability. And then if you let the system go and you allow the system to make changes then from this point off, you'll be able to, you'll be able to start to see an information accumulation, which I think is what I will consider as being now we have a living system. Again, we have not done this yet, but I think this is a way to look at it and get a sense of how to view these things. And again, once you believe that, then of course, what one can do, one can look at a chemostat, and we can put the system, the equations in, and then we can determine, we can solve. This is easy to solve, and we can find the transition to life that actually depends on the rate of course, it depends on we have to have the formation rates are faster than the decay rate, and it happens that this is how fast you thin out the system. This is I mean 1 over tau, the time here, and this is the in rate of resources, and this is a hydrolysis and of the what's it called, of the system. Let's see, this is resource hydrolysis rate. Yeah, this is where we have at this is the integration of the two, where we integrated the rates for replication and the charge transfer. So just want to sum up here by mentioning, I mean, I didn't have time to go through all of that, but I'm so excited about it because we've just finished a study where we crank our way through and determine every single free energy change for every reaction in our system. And we came up with this matrix here on top, where the ones with yellow, that's the input. And some of the interesting, and then once you have all these things, then you know more or less, if you want to say have a system that has a diameter of 1 micron, similar to maybe a E. coli bacteria, then you can figure out how many fatty acid formations you have to make, how many information replications you have to make, how many vesicle self-assembly processes you have to account for, and also how many self-assembly processes you have to account for with where both the metabolic, I mean, both the ruthenium complex and the self-assembly, I mean, the DNAs, they anchor into the aggregate. And we were surprised when we actually calculated this through, and we say that we have this hierarchy. So about 97, or this is not surprise, 97% of all the free energy changes in the life cycle is for metabolic processes. Only 3% of the changes has to do with self-assembly, and the vesicle fission energetics is completely out of the picture, and it's very, very, very weak. These bending energies are orders and orders of magnitude smaller than the self-assembly energies. I mean, I have not, well, one of our collaborators, he knew that, well, the rest of us didn't. So anyway, so then we compare, there's a recent study by Ortegas or Sola that actually looked at an E. coli 1 micron, and they found that this is how much a life cycle takes in terms of energy input. This is how much our life cycle takes from the protocell, and it's a little less than 1/10 of an E. coli. I thought there was a much bigger difference. Another thing that I found kind of interesting is that the efficiency, metabolic efficiency, is about 300 joule per gram, both for our system and for a modern cell. And both our system and a modern cell uses by far most of the energy to produce the vesicle, to produce the container, about 99%. So that was it. Just to sum up, we talked a little bit I compared a little bit with how I imagined that life could emerge on the Earth, and how we as humans could actually make a living system in the laboratory. And then we've discussed in particular connection between the energetics and information, and We've looked at kinetics of charge transfer and information replication. We've looked at thermodynamics of every protocellular process to get this energetics hierarchy. And then we discussed here at the end also a little bit about the characterization of transition from non-living to living materials. So thank you for your attention.
So thank you very much, Steen, for this wonderful presentation. Now it's time for questions. If you have, yeah.
Okay, thank you very much for being here first, and congratulations for the amazing amount of quality of the work you have been doing for years. And I have a question regarding your model, the experimental system you have developed. Are you planning to consider also kind of channels inside the membranes? I mean, to allow perhaps the movement of some of these components from the outside to the inner part of the protocell, or to allow some kind of flip-flop mechanisms that could allow DNA to go inside, so that perhaps the evolvability of the whole system could be improved, and also in this connection between artificial life and real life on Earth?
Yeah, I like that question. I mean, it is kind of odd. There are not many in our community that look at protocells where everything is on the outside. But there are Masayuki, he has made experiments where there are ways by which you can flip. You can simply turn a vesicle inside out by changing the environment conditions. And yeah, later on, obviously that happened. I don't know whether it happened with channels, so you can actually have flipping in that way, or it simply happened by inverting the system. But again, yeah, I mean, we're doing this, and it's difficult enough, takes all our time and energy. But I think these are really interesting questions, and I would love to work with RNA instead of DNA, because I'm sure there'll be more people excited about it, but it's just so much more difficult because the RNA deteriorates, and they also pretty much all the charge transfer studies, which is characterized for years, is on DNA, and very little is on RNA. So that's why. But yeah, I would love, maybe you guys, there's some RNA people here, right? This is something you guys should do.
Okay, thank you.
Yeah.
Okay. Thanks for this very nice talk. I find very intriguing these last results of you about the efficiency of the difference in efficiency between the protocell and the E. coli.
Ah, yeah. Okay.
And I was wondering if that means that real cells are operating very close to the optimal efficiency, or is just, do you think that it could be, or just...
I think it's a little bit of we were all very surprised about the efficiency, and because I would think that such a dumb design as ours would be very inefficient. But I think why we got a little bit cheated, or why it comes out like that, is because, let me see where it is here. Is if you look at these molecules here, there is very little difference. I mean, they're very nutritious input we have. There's not much difference between what we put in and what we end up with. And I think that's why the efficiency is high. If you have to go through a lot of tedious steps where you build up more complex molecules, that takes much more energy. And that's what modern life has fine-tuned all these with catalysis and with enzymes, so it runs like a spotted dog. And of course, we can't do that with prebiotic chemistry without enzymes. So I think the reason why we're getting this high efficiency is because our input is pretty sophisticated, and the E. coli can eat the sugar and peanut butter, and it can eat all things. Good question.
More questions?
Thank you very much, Steen, for being here. I wonder if the students are aware of how inspirational your work has been for several generations of physicists like me and many others that found in biology and in the origin of life in particular, a nice place that we had not been aware of during the degree, that we could apply all the theory. And well, for the last 30 years, I think in particular, I mean, perhaps you don't know, but your core world, he was one of the first people who were able to use computer models where actually different programs could compete for the memory. And that would be a very, very nice metaphor of life. So it's one of the first great ideas, I think, of how computers, not only by models, but also from another point of view, could shed light on how life can be modeled from many different points. So thanks for that. That way, we are very, very happy that you're here. And my question would be more general than the other ones. So you're working on how, what should have a minimal model for life and what is not so important. So in your opinion, after all those years of work, which do you think would be the main differences between Luca and your model?
Which one?
Between what you've been trying to do, like the smallest cell that can work. Which is your opinion of how should Luca be? So which would be the main differences between what really happened and what we are trying to model now? So where are the open questions and which are the questions that we really don't know?
Yeah. Well, first of all, thank you for your kind words. And actually, as I was preparing here, I didn't quite know whether I should take some of these, these early computational things in here. But I thought, since you guys are astrobiologists, then I thought it was better to keep to the good old physical chemistry. But yeah, so what is the difference? I don't know. I mean, I know. I actually think this one works also for modern life. I mean, you have a metabolism, and it makes these things. And this is what it spends most of its energy to, and it certainly makes it replicates the information. So if you just look at it as a chemist, then the energy transducer and the cofactor, that is the catalyst. Also in modern life, even though you usually don't look at it, but if you go far enough away, that's what it is. So I think that this one holds for modern life too, even though modern life is much more sophisticated. However, how we get there, I try to hand wave a little bit, and I don't know how something like this could be turned into. There's so many things, there are new materials there, as you also indicate, we have to get all the good stuff inside the vesicle, and I simply don't know. I don't have any qualified guess. I mean, I could probably some of you that know more about that. So we've been focusing on trying to get to bootstrap to the point where we have a system that can take in resources and then use free energy to make building blocks, so the system can grow and divide, and then have information that at least in part controls this growth and division process. That's sort of our operational definition of physical chemical life. And I mean, it's amazing. When I was a young man, I thought, if I just get a grant in 5 years, we'll do it. And then I became a little more realistic. I started to work with chemists, and he said, okay, no, you cannot, you need much. But I said, okay, 10 years. Now, it's taking a long time. It's very difficult. Well, actually, it's more difficult also to get money than it is to actually do the science. So as you all know, that are a little older, that getting money is for this kind of stuff is really hard, because it's hard to. Well, it's getting a little easier, I guess. I hope with all this AI, because suddenly you have AI that actually has that works, and I believe that. Once we have self-replicating, I mean, artificial living materials, you can make technology that can do all kinds of fantastic things, because you have the power of life to make your technology. And now that, well, at least that's what I hope, that people will be more open to that. But so far, I mean, well, I've been lucky and got a lot of money from time to time, but we are fighting ferociously to get another grant to hopefully, now get the last piece. Else you guys, maybe some of the young people want to go in and finish the work and show how it should be done.
Thank you for your answers. So are there more questions in the room? No? And in Zoom? No. So I just wanted to ask also a very general question that is related with what you were talking about, that is just with your experience, how far do you think the scientific community is of creating an artificial living system?
Yeah, well, we are closer than we were when I started, but I had no idea that it would take that long. In the beginning, I was really scared that some other group would create life before us. I'm not so scared anymore, and I think that there was a period back in the early and mid. In early 2000s where there was a lot of money flowing this way, but then somehow fell out of fashion for reasons I don't know. But I think it's,
it's more complicated than
yeah, it's more complicated than I think or that I originally thought. I mean, I think that there's a bunch of groups that are very close to doing it. There's many people there are many more people working on where they have what's it called, a translation machinery, and then they are sort of picking from modern cells, and then they are making things that are more cell-like. I would probably put the money on that, but even though the Dutch they got a lot of money several times, and they still have not done it. The other problem is also to manage such a big project, because you need as you've seen here, we started different. We said, hey, this is a concept, and which chemicals could work inside here. Because theories can work, we can do the equation, doesn't matter what the chemical is, but for chemists, it takes years to be an expert in particular material. So once you get some world experts to work on these things, and it turns out, you actually have to modify this because else it doesn't fit with the other things. Then you know how fickle it is to get funding for people to, no, I'll not risk my life and livelihood, so they don't want to change. They continue to do what they're good at, and so they can continue to get money. So then we also have a situation which is makes it structurally difficult. That's where Los Alamos was great, because Los Alamos, in the old days, they made bombs, they made the atomic bombs, that they know how to make big projects. So there you go in, and everybody pools their resources to do one thing. That's very difficult to do in an academic environment. But then we've talked to private investors also, and they're really excited about this, except when they realize that there aren't really any killer applications. We can't go out and make billions of dollars or euros on this. So I don't know. I'm open for any suggestions for how we can get further.
Okay, thank you. Very interesting.
Oh, can I just ask one thing? Do you guys agree, or how many agree with this way I talked about life, where we included the metabolism? I started out with this energetics. Is that needed, or is it not needed? First, who believes that it's needed to find life? Yeah, but I'm not saying. And who thinks that we can, if we just have a self-replicator like a virus, that's enough? Is that virus alive?
That's very controversial. Yeah.
Yeah, because it's anyways. We have at the upcoming Artificial Life meeting, we have a workshop, special workshop dedicated to 3 hours, where we try to actually see if we can define life. Because in the beginning of the Artificial Life movement, there was a lot of discussions, but it was all theoretical, and we got sick and tired of it. So now we have to shelve that until the community has made some progress. Now it's 35 years later, and I think that there might be good, suggestions for how one could define life. What do you think?
Yeah, I fully agree with you. I think that in the framework of prebiotic systems chemistry, unless you combine a compartment with replication and metabolism, you don't have life. So I think these three prerequisites should be somehow combined.
Yeah.
If you want to recreate artificial life, or if you want to explain how life could have started 4 billion years ago.
Yeah. Okay. Okay, but I hope that there was some disagreement, so we could have some. Anyway, thank you so much for coming. Yeah.
Thank you, Steen.
Thanks for the talk.